So I'm going to get started. So my name is Cibu Thomas. I am a staff scientist in Dr. Chris Baker's lab, the section on learning and plasticity, and I'm going to be talking about imaging changes in brain anatomy. So first off, just to give you a sense of the scope of this lecture that I had in mind, what I'm going to talk about basically is a cognitive neuroscientist perspective on imaging brain changes. So if you're starting a project focused on studying structural brain changes, or you're in the middle of a project, I think this might be useful for you. Um, while the bulk of the talk is entirely about imaging structural changes in the brain, I think it extends to also like looking at functional changes. So, as long as you're familiar with certain kinds of MRI images and to some extent the MRI jargon, I think you should be fine. If at any point, if you have any questions, feel free to stop me, and I should be able to clarify them. All right, so just about everything changes the brain. So starting from infancy, even in the growing fetus, things are changing rapidly. When you learn stuff, your brain changes. When you're acquiring new skills, your brain changes. Sleep habits change the brain, mood changes the brain, stress changes the brain, nutritional factors change the brain. So just about everything changes the brain. The reason why imaging or measuring brain changes is particularly important is that, one, if we can properly measure this and understand it, it can potentially help us slow the effects of aging or some disease. And if we can measure the efficacy if we can measure this properly, then we can look at how certain treatment strategies can help or should be stopped. And from a larger perspective, it can also help, for instance, guide public policy on promoting good mental health. So imaging brain changes and sort of understanding what are the mechanisms underlying brain changes is very crucial. So how do we study the changes in the living human brain? So you can think of the kind of changes into two dimensions. One, in terms of the temporal dimension, you have changes that take place in the scale of years to months, weeks, days, or even seconds. But in terms of the spatial dimension, you have changes that can happen at the level of the whole brain. In some cases, it could be in the scale of centimeters, millimeters, or even microns. What makes MRI a particularly powerful tool is that in terms of the spatial dimension, it allows you to look at both the whole brain level as well as at the level of millimeters and more recently even at the level of microns. So the things that check off really well with MRI is that, one, it's safe and non-invasive. It's also translational in that anything that you develop in an MRI scanner can be then taken to the clinic, in a hospital. You can deploy it to treat patients and so on. And probably one of the really cool things is that it's multimodal. And what I mean by the multimodal part is that it's the same device, but you can sort of sensitize the device to look at different tissue properties. So what I'm showing you here is a T1 weighted image, which gives you very nice gray, white matter, and CSF contrast. So there's white matter, gray matter, and the CSF is black. But with these images, you can then compute things like the volume of a given portion of the cortex. You can look at cortical thickness, gray matter volume, gray matter density, and so on. Now, you could have the same subject, the same device. You could tweak the sequence and make it sensitive to deoxygenated hemoglobin. And now you're looking at task-driven fMRI, or you're looking at resting state fMRI, and you can look at functional connectivity and so on. Tweak the sequence and make it sensitive to the diffusion of water molecules, and now you can look at measures of tissue microstructure. And also look at measures of major fiber pathways in the living human brain. Now, in terms of measuring structure, you're going to do all of this, right? Now, in terms of the temporal dimension, you can now have a whole host of behavioral methods from which you can look at changes in the scale of years, or milliseconds. So it could be a reaction times task, or it could be like even a questionnaire looking at how someone's mood is improving with treatment or some other intervention. So with these two things, the change in behavior and the change in structure, you can then look at the correlates, what is driving each other. So the outline that I have for today's talk is to review some of the popular MRI methods that are used for measuring brain changes. Now, what I'm showing you here is some of the more recent methods, like the quantitative T1, quantitative T2, including very classical methods like T1 weighted images, as well as diffusion weighted images. So for the purposes of this talk, I'm going to focus mostly on T1 weighted images, as well as measures or maps that you can get from diffusion weighted images. But the sort of ideas that I'm going to talk about extend to just about any structural imaging method. So I'll review these MRI methods, and then what I thought would be really useful was to know the limits of these techniques. My previous advisor used to say that the devil is in the details, so you should know the devil. And in terms of acquisition, data processing, inference, and all these things. And I'll also sort of give you an overview of how, over the course of years in doing this kind of work, I have learned how to overcome some of these limits, and also briefly talk about the future of imaging changes in brain anatomy. So let's first review some of these methods. So as I mentioned, T1 weighted images, these can be acquired in less than 6 minutes. It is a qualitative image. What's really nice about it is that it gives you very nice tissue contrast between 3 tissue types, the gray matter, white matter, and CSF. In the clinical domain, with just this quick picture, the radiologists can quickly diagnose lesions and so on. And it's profoundly useful in the clinical domain. What we can do in terms of research is take these same images and use software packages developed by neuroscientists and sort of skull strip, segment these into different tissue classes, parcellate them into an atlas, into different lobes, different subcortical structures, and so on. And from this, you can then compute subject-specific measures of the volume of cortical and subcortical areas. You can also extract higher or second-order measures. It's all from the same data. But using these same methods, now that you know, for instance, the gray-white matter boundary as well as the gray-PIA boundary, the PIA matter boundary, you can then compute the linear distance between these two boundaries and call that as cortical thickness, and you have a measure of cortical thickness in vivo, right? You can use these same measures and compute things like gray matter volume in different areas of the brain. You can also measure things like the height of the gyri, the depth of the sulci, curvature measures, shape measures, and so on. And with this sort of approach, there's been a lot of studies that have reported how there are longitudinal changes in cortical thickness associated with aging, with clinical conditions in autism relative to typical development, and also things like training, how memory training can change and evoke changes in cortical thickness. So that's how, a quick overview of T1 weighted morphometry. Diffusion MRI is quite different, and it has certain strengths that other MRI methods don't have. So here is a standard T2 weighted image, 3 hours taken 3 hours post onset of a stroke. And what you see here is a chronic infarct in the subcortical white matter. If you take a diffusion weighted image of the same subject in the same area, what you see is, unlike the T2 weighted image, you can see how much the infarct is extended. So it is very sensitive to the diffusion of water molecules. Here, with the stroke, you're seeing an attenuation of the diffusion of water molecules, and you see this really bright, and this allows doctors to intervene rapidly. But the other cool thing about diffusion is that, so this again is the T2 weighted image. Now, all you can see is basically just the white matter, gray matter, and CSF. But what diffusion can tell you is this. it actually allows you to sort of parcellate the different white matter structures. So here, red is showing the direction of fibers in the right-left direction. Sorry. And let me just do this again. Green shows anterior-posterior orientation, and blue is inferior-superior. So you have the unprecedented ability to look at the orientation of white matter pathways in the living human brain. The way it works is, very briefly, if you imagine each one of these things as water molecules, in a restricted environment, like let's say a cylinder, which is what we think of an axon, you have the diffusion displacement mostly in one direction. So you can characterize this diffusion displacement in the form of a diffusion tensor. And these three different diffusion coefficients, from these diffusion coefficients, you can extract these really cool maps. This is fractional anisotropy. This here is a directionally encoded color map that I briefly talked about. This here is the mean diffusivity or trace. So you're using basically these three different diffusion coefficients, and you're computing different maps that can tell you different tissue properties. This here is axial diffusivity. This is just the dominant diffusion displacement. This here is the radial diffusivity, which gives you a sense of how much diffusion is restricted perpendicular to the direction of the axons. And you can make inferences about whether a structure is myelinated and so on and so forth. The other cool thing about DTI, or diffusion imaging, is that, so in this same diffusion-weighted slice, if we look at the diffusion displacement profile in this particular region, I'm going to expand this, in the CSF, it's likely to be isotropic in that the diffusion of water molecules is fairly homogeneous. But inside white matter pathways, it's highly anisotropic, so it's heavily restricted. And so you can characterize the diffusion displacement in this manner. So these tend to be spherical. These are highly prolate. And from this, using computational algorithms, you can just string these together to then look at or image white matter pathways. And from this, you can compute measures of axial diffusivity in a particular tract in the brain. So there are now several studies that have used these kind of techniques, and they've looked at how with brain development there is changes in brain wiring, longitudinal changes in the corpus callosum with traumatic brain injury, and also training-related changes in the brain following learning novel tasks. So that's like a quick overview. Now I'm going to talk about the limits and the devil in these techniques. So the traditional approach to looking looking at brain changes is predominant to use a longitudinal design, purely because it provides you the strongest argument for making a causal link between some intervention and the change that's been presumably evoked by that intervention. So the standard approach then is like you take two groups of subjects, an experimental group and a control group. This here is a T1 weighted image. You look at let's say you have some pixel intensity here between the two groups. You give them some training, let's say it's juggling, and you're sort of assuming that there are some structural changes, possibly in axons, blood vasculature, and so on and so forth. And after all the training, you are going to image them in exactly same scanner and so on. And the hypothesis is that the training is going to change relative to the control group. There's going to be a significant change in this training group in a particular region and so on, right? And this is usually tested using a task by, it's like a group by time point interaction, where group is like control and experimental group, and the time point is pre and post. Now, if you look at the MRI literature, what we found is that the evidence for MRI-based studies of brain plasticity is not so robust. So what we did was we looked at animal models, which predominantly seem to provide the most compelling evidence for training-specific changes. So in this particular task, and here's a classic example. So in this particular task, these mice were trained to retrieve a food with a particular paw, right? And so like there's a pasta, and they have to use their paw in a certain way in order to retrieve that particular food reward. And what they showed was that within like a day, relative to a control group, the training group show these changes in the dendritic spines. There's a massive proliferation of dendritic spines. So this is not MRI. This is two-photon emission two-photon imaging. And so you have the spatial resolution of like 2 microns, and you have the ability to actually longitudinally image changes in something as small as dendritic spines. But what they were particularly, what makes this really compelling is that relative to a control group, which is in white, this particular study, they showed that the change is specific to the training group in the contralateral forelimb motor cortex. So the trained motor cortex showed a proliferation of dendritic spines, unlike in the trained group in the region outside the contralateral forelimb motor cortex, which is in gray, as well as training in the ipsilateral motor cortex. So what they managed to show was that there's a specificity to the training group, and it's specific to the brain region. And they also looked at the same animals that failed to learn, and they were able to show the specificity to task. And finally, they also looked at the relation between the number of successful reaches and the change in spine formation. And so they could correlate this change in structure to a change in behavior. So we used this as a framework for assessing the robustness for training-dependent structural changes. So the way we were looking at it was that if you pick any study that is reporting a change that's evoked by some training, so you could ask the question, to what extent is this change specific to a group, to the training group, specific to the task? If the claim is that it is specific to a brain region, to what extent is that true? And does it correlate with behavior? And so we used this as a framework, and we looked at the existing literature, and what we found is the bulk of the evidence was not very compelling. The problem is that in terms of MRI, there are several sort of challenges, and it's in terms of scanner-related issues, sequence-related issues, subject-related issues, and so on. So what do I mean by this? So in terms of scanner-related factors, if we were to use a ruler for measuring a change in cortical thickness, no matter where you use it, this is a standard measure. You're probably going to get 2 millimeters plus or minus a couple of microns. And that doesn't matter whether you do it here or in Pittsburgh or in Mongolia, right? But if you use a scanner, if you use the same exact scanner between 2 different sites, you're going to get different measurements. So there's intrinsic measurement error that is problematic. Now, this is not a problem when you start a project in, let's say, NIH, because you're going to stick to a scanner and you're going to have the same sort of stability to it. But if you are going to use data from, let's say, the Human Connectome project, where you have data acquired from different sites, this is something that people need to be aware of. The other issue is the sequence-specific factors. So let's say a T1 weighted image, we're often confronted with the question of, okay, do we use acceleration, for instance? So acceleration gives you faster acquisition, so you get an image in less than 6 minutes. But the contrast to noise of a T1 weighted image with acceleration is much less than what you would get with a 10-minute without acceleration image. So here's an example of that. So if you look, unfortunately, these colors are sort of weak. But what I'd like to point out is over here. The distance between the 2 lines here is quite different with and without asset, which is the acceleration factor that you use in a GE machine. And so this can result in systematic differences in your measurement of cortical thickness. So what we can do then is to use either 2 sets of accelerated images, because in a pediatric population, you want to keep the subject you want to get these data quickly so they don't move so much, because these sort of images, once the subject moves, then the entire scan is wasted. So if you want to keep the scan short, then you can use an asset, get two sets of asset images, and you average them, you get better images. So so the question as to whether you should use acceleration or not is something that you want to consider before you start a project. Depends on the pediatric, whether it's the kind of nature of the project, the kind of patients that you're using, and so on. So in terms of DTI data, one of the problems that we often see is eddy current distortions. So these are sort of high, like what you see is intensities in the periphery of the brain. These are caused by the rapidly switching diffusion gradients, and it distorts these images and alters the diffusion sensitization, particularly in the periphery. But what happens is that if you don't correct for this, these look like high anisotropy, and depending on the anatomy of the brain, you get extremely profound differences between subjects. And this could manifest as training-related changes or some sort of pathological changes. But these are all correctable if you use the latest version of FSL or Tortoise, which is a package that's developed in the NIH. You can really do a really good job of removing these sort of distortions. The other common artifact is called Gibbs ringing. So this is a movie where what I'm showing you is these high intensities, particularly in regions where there are sharp image transitions between the gray matter, white matter, and CSF. And this, again, can manifest as high fractional anisotropy, particularly in certain brain regions, and can be misleading. Again, this can also be corrected, particularly if you use a package called Tortoise. The other very common distortion is the EPI distortions. So what I'm showing you here is a T2 weighted image, a slice of the brain of the same subject, one at the level of the orbits of the eye, and this is a little superior. Now watch what the diffusion images look like. So this is the same slice, but you can see that the orbits are completely warped. This is if you acquire it in the anterior-posterior direction versus the posterior-anterior direction. So you can see that there are signal intensities here, there are signal pileups here, as well as distortions. The entire brain structure is warped. And so it depends in what direction you acquire the diffusion-weighted images. But then again, you can correct for these sort of distortions. What you need to correct is both a blip up and a blip down sequence. Basically, if you're collecting an anterior-posterior phase encode data, you need a posterior-anterior set. And with a T2 weighted image, there are ways to correct it. The reason why correction is important is if you're interested in doing tractography and looking at changes in white matter pathways, here is what the tracks look like, the corticospinal tract, for instance, in data that is not EPI distortion corrected. So you look at it, there's the corticospinal tract sort of ends prematurely in one hemisphere, but once you correct it, you get much more anatomically truthful looking corticospinal tract. So those are sort of scanner and sequence specific or sequence related factors. Now, there are other subject related factors. So one that pretty much everyone is aware of is subject head motion. So here what I'm showing you is changes in gray matter volume and cortical thickness that can be measured based on just how much a subject moves his head. So this study by Reuter et al. Showed that head motion impacts both gray matter volume and cortical thickness estimates, and roughly 2 millimeters per minute motion can result in a roughly 1.4 to 2% loss in gray matter volume. When there is absolutely no atrophy or no change in gray matter volume, this can manifest as a gray matter volume change. So you can imagine that a subject group that is likely to be less comfortable in an MRI environment can systematically produce motion artifacts. And if we don't correct for this, we can then interpret this as a gray matter volume difference when there is very little data to suggest that. Now, how do we correct for this? With a T1 weighted image, it's really hard to correct data that has motion in it, but we can use other techniques like prospective motion correction, as well as much more old-style techniques such as better padding and putting the subjects through a mock MRI scanner, making them comfortable, and so on. The same sort of problems affect diffusion-weighted images. So here, this is the directionally encoded color map before correction, and after correction, you get reasonably looking directionally encoded color maps which are faithful to the anatomy. There are other factors also. For instance, blood pressure, for instance, can have an impact on T1-weighted images. So here, this is a study by Scher et al., which showed that these dark spots are basically regions which presumably showed a change or a reduction in gray matter volume in a group that showed higher blood pressure compared to a group that showed normal blood pressure. So this also tends to have an impact on gray matter volume estimates as well as cortical thickness. Similar sort of physiological factors can also have an impact on diffusion-weighted images. So this is what a directionally encoded color map, as measured using DTI, looks like at the diastolic phase of the cardiac cycle. But in the systolic cycle, you see basically a disconnection in the corpus callosum. You can see there are dramatic changes in the white matter anatomy in the systolic cycle. So controlling for this can be done using cardiac gating and so on. But the basic point is that there are other reasons why you would find systematic differences in fractional anisotropy and other measures of DTI that DTI produces. So if you're interested in, for instance, looking at how mindfulness meditation or yoga, for instance, can change brain anatomy, this is something that you need to be careful about, because what changes with mindfulness meditation, for instance, is potentially the respiration as well as your blood pressure. And this can manifest as a change in fractional anisotropy. It's purely physiological, but it might be quite unrelated to the white matter. It could be mostly physiological. So these are things that you want to control for. So you want to measure and show that there are no systematic blood pressure differences or respiration differences between the two groups. What I'm showing you here is changes in fractional anisotropy. It's a measure of structure that is changing with changes in the cardiac pulsation. So these are regions that are under very high risk of showing a difference in fractional anisotropy purely because of changes in the cardiac cycle. So those are subject-related factors. The other things that you probably need to know about is that even the version of software. So this is a paper that came out in PLOS One where they looked at different versions of FreeSurfer, the workstation type, and the operating system so you're using the same data, but you're processing it using different versions of software. And it can produce significant differences. The reason being that certain operating systems have a certain floating point correction, and that when you do it iteratively, produces systematic changes, and then lo and behold, you have some changes in cortical thickness measures. So the solution is to pick a stable version and then stick to it. So if you are starting a project, stick to that particular version and don't change. There are other factors like data processing factors, and what I want to show you here is that this is probably, in terms of T1-weighted images, this is probably the raw data that you initially get. But what the final product of all the analysis would look something like this. What's happening in between is all these steps. So there's normalization of these images, segmentation, modulation of the gray matter, and smoothing, and so on. So there are several transformations that the data is undergoing. And at each point, it's sort of like a recipe. And so biases can be introduced. And for instance, one study showed that depending on the filter size that you use, the smoothing kernel that you use, you can take the same data, use different smoothing sizes, and you could get completely different statistical maps, and you can conclude that one group is quite different from the other group because of these three in these certain brain regions. And it has very little to do with the anatomy, but it's purely because of the way certain parameters were chosen. The other interesting study that I found was from Adam Thomas, where what he did was he trained a group of people to become experts in a visual motor task, and he found clear behavioral evidence that they were indeed doing really well in this task. He also did functional MRI and found that there were changes in Bold signal that were predictive of or correlated with the change in behavior. And so he looked at structural changes. And what he did was he compared two different packages. So here's what the SPM approach showed, changes in certain brain regions. But if you take the same data and run it through a different pipeline, he got a completely different map of structural brain changes. So this just goes to show that in each of these packages, there are several assumptions being made about how the data are processed, and each of these things can have an impact on how your final maps can look like. So the easiest way is to talk to someone who's done this, particularly in terms of using statistical methods, your local statistics guru, and they might be able to help you at least understand what are the sort of inference sort of assumptions being made in when you use SPM versus like FSL and other approaches. So to summarize this part of the lecture, so basically, if you're starting a project, pick a stable scanner and stick to it. Pick a robust MRI sequence. There are, if you were interested in a T1 weighted sequence, people like Vinay can help you pick a sequence, walk you through what are the sort of parameters that you need to really be mindful about. Also, if you're using a diffusion study, it's worthwhile to sort of think in advance about what brain regions you're interested in, because the kind of parameters that you choose, like for instance, in which way you would acquire the data in terms of the phase encode direction, can also help you pick a particular parameter which can help you better resolve, let's say, the frontal lobe versus the brainstem. So if you're not so interested in the brainstem, you're better off doing like an acquisition which is more tuned to looking at the anterior-posterior phase encode direction than the right-left phase encode direction. What I've learned over the years is that better data comes at a cost. You need more time. So if you're interested in looking at brain structure, earlier, what people used to do, and that includes me, we would just do a functional imaging study and tag a 10-minute structural image sequence and use that as our basis for studying structural changes. But over the years, what I've learned is that if you're interested in structure, do 1 hour of structural imaging, and that's going to pay off. There are all sorts of potential confounds that one needs to be mindful about, particularly head motion, physiology. And it's also worthwhile to just pick a stable pipeline in terms of FreeSurf or for surface-based analysis, or if you're interested in voxel-based morphometry, FSL or SPM has now much more stable pipelines. If you're using diffusion data, use Tortoise. It's been extensively tested. It's developed in the NIH community. So if you have issues with how the data is being processed, you can go and talk to the people who developed it. And the other piece of advice that I have is to QC images as you collect them. It's really hard to recollect these images, so even as the images appear on the screen, keep an eye on it. And when you look at these images much later, when you're processing these data, look at each slice. There's no compensating for it. It is like we have these packages where you can just like write a script which will do everything from reprocessing to taking it to the statistical maps, but it's really important that you look at each output at each stage and take a magnifying glass to it. Also, I think it's really it's important to be consistent with the instructions to the subject. These things matter. So if you're going to do, even though it's a structural scan, I think it's really important for you to be consistent with with the way you're instructing each subject. So having talked about all these sort of issues or challenges with imaging brain structure, particularly in looking at brain changes, here's a study that I did a couple of years ago. And I mean, I'm taking all of these lessons that I've learned over the years, and I'm trying to apply this to this particular project. And I wanted to share with you how this worked out, right? So the question that we were interested in was how does learning versus mastering a motor skill change the human brain? So going by sort of the framework that we were talking about, we wanted to show that any change that we evoked was specific to the task, specific and correlated with behavior and so on. And so we designed a study where each subject, this is a longitudinal study, and so each subject would come over multiple visits. We scanned them at, let's say, 10 in the morning for 2 hours, gave them 2 hours of rest, and then scanned them again for 2 hours. And so for the first visit, it was a baseline scan. In visit 2, we scanned them in the morning for 2 hours, and then we gave them a right lateralized visuospatial training video game task. The thinking was that if you right lateralize it, so any change that you are expecting would then, at least to some extent, would be lateralized to the contralateral hemisphere. And then we, immediately after the training task, we imaged them again for 2 hours. 1 week later, we got the same subjects back. We scanned them for 2 hours. This time, we trained them on a left lateralized motor sequence task. So what we have here is a rest control as well as an active control. So you can look at how motor sequence training can be is different from, let's say, visuospatial training and relative to a rest control. So there are 2 controls, an active control and a passive control. And then in visit 4, the difference between visit 3 and visit 4 is that after visit 3, for each of the weekdays, and so these are a week apart, during each of the weekdays, we gave them the same training for each of those days, 1 hour of training in the same motor sequence task. So this was our attempt to look at sort of acute changes immediately after training, as in short-term training, versus long-term training. So all this repeated training, to what extent does this change brain structure? What we found, however, was the most dominant effect was that just time of day, AM to PM, has an impact on surface-based measures of the brain. So in terms of cortical thickness, what I'm showing you is that there is a change in the magnitude of cortical thickness in these brain regions purely from AM to PM, even without any task. So these are regions which show statistically significant differences in cortical thickness from AM to PM. Now, if you do the same analysis with the same data and look at, use a voxel-based morphometry style and look at gray matter volume changes, again, you see, without any training, you can see changes in gray matter volume in these brain regions, right? So what we've identified here is that a simple confound from AM to PM, there is a significant change in apparent brain volume or cortical thickness. Now, because we had different kinds of MRI data, so we had diffusion data from these same subjects, we then looked at to what extent is this explained by the diffusion data. So looking at mean diffusivity, what we found was that from AM to PM, there's an increase in the diffusivity of water molecules in these brain regions. So there is the diffusion of water molecules is basically increased in these brain regions, particularly along the sylvian fissure, the interhemispheric fissure, and so on. We had in our sequence a particular sort of manipulation that allowed us to then look at to what extent is this change in mean diffusivity explained by changes in free water, right? The perfusion, sort of CSF-related changes in the brain. Now, when we looked at it, what we found was that, again, in these same brain regions, the bulk of the change in mean diffusivity, the increase in mean diffusivity from AM to PM, was explained by an increase in the free water volume fraction, suggesting that this is probably something that's related to changes in cerebrospinal fluid from AM to PM. Next, we had resting state functional connectivity data, and we looked at changes in functional connectivity from AM to PM. And again, what we found was that without any training, from AM to PM, there's a systematic increase in functional connectivity in these brain regions. This is, if you seed the hippocampus and look at functional connectivity with the rest of the brain, you see these regions showing an increase in functional connectivity from AM to PM. If you take the sensory motor cortex, bilateral sensory motor cortex, see that, and look at changes in functional connectivity from AM to PM, you see both increases and decreases from AM to PM without any, which is very little to do with training. So just time of day seems to have an impact on these maps. Now, what I'm showing you here is an overlap of all these different modalities, right? So this is the blue areas show you the decrease in gray matter volume because of time of day. The pink areas show an increase in the free water volume fraction from AM to PM. And the green areas show a change in functional connectivity from AM to PM. And as you can see, there is a fairly nice overlap in certain brain regions, which all points to the possibility that this is a physiological effect. So in one sense, the study that our goal was to look at training-related changes, and what we found is that some of the, like at a much more fundamental level, there are just doing nothing can evoke a change in the brain and the MRI is actually sensitive enough to pick up these changes. So what about the impact of training? So we then looked at the cortical thickness maps, and what I'm showing you here is the interaction between the time point, AM to PM, versus the different visits. So the visuospatial training versus the motor sequence training and so on. And we do get regions which show changes and increases in cortical thickness. However, these clusters do not survive correction. The only thing that survives correction was, and it's barely visible, is the correlation between change in cortical thickness as well as a change in performance. So, so what I'm showing you here, this outline, is the region which we used, for which we used a functional localizer to identify the regions that are activated when subjects perform the task. So it's a functional localizer. And this is one of the regions which are activated while performing the task. And then in this particular region, you get like a really small region that is showing a positive correlation between an increase in cortical thickness and a change in the performance and a better performance. But what's interesting is that while you see an increase in cortical thickness correlating with better performance, when you look at long-term motor training, a decrease in cortical thickness is correlated with better performance. So one possibility then is that with cortical thickness, immediately after training, you're seeing some changes in the T1 signal. Again, it's really hard to interpret this as a change in cortical thickness because changes in blood flow, because of the activity that the subjects are performing, can change the T1 signal, and it can manifest as a change in the gray matter signal, and which in turn can be interpreted as a change in cortical thickness. But it could be a change in blood flow, for instance. Now, the same thing can manifest as a decrease in cortical thickness, because if you have an increase in white matter, the border between the white matter and gray matter boundary is now going to shift a little up, and now you're going to have a decrease in cortical thickness because that distance is reduced. And that can correlate with behavior. So what I wanted to point out is that, yes, you can get compelling evidence for training-related changes, but the interpretation of this in terms of mapping it to some sort of biological change is not very straightforward. Now, if we take that same data and run it through the VBM, or voxel-based morphometry pipeline, and you look at correlation between change in gray matter volume and change in performance, we do see some significant changes in the trained hemisphere. However, this is in volume space, but it's not really, it doesn't overlay well with the surface maps that we saw. So there is something interesting going on, but it's not very trivial to actually nail it down to saying, yes, this is strong and compelling evidence for changes evoked by training. We did a similar analysis with the diffusion data, but we couldn't find anything that was compelling enough. So what I have learned from this is that in terms of the best way to sort of disambiguate changes that are evoked by confounds versus the actual experiment of interest, what matters then is the experiment design. It can help you sort of tease apart what is likely to be a confound from what is likely to be a real effect. So in our case, the rest control helped us identify the time of day as a significant confound. But what's interesting is that MRI measures are sensitive enough to detect this effect, right? So from a design perspective, we find that this is really important, not only in the context of a longitudinal design. So if you're looking at brain changes in a training study, you want to scan these subjects at the same time as you scan them pre-training, post-training as well. Even in a cross-sectional design, this is particularly important because, for instance, purely for pragmatic reasons, you might take a group of healthy volunteers just because they are available, because healthy volunteers probably are engaged in some sort of activity in the daytime. You probably end up scanning them after 6, while your clinical subjects might be available during the daytime because parents can bring them over. So it's not something that you chose, but you're introducing a systematic confound which can then manifest as changes in brain anatomy, but they are probably not so. And as I've shown, a confound like the time of day effect appears to be impact across different modalities of MRI, which suggests that this is not just a confound because of the methods that we used or the MRI sequence or anything. It's a physiological effect. And because we had multimodal data, like we had the diffusion data, we could tell why we saw a decrease in gray matter volume. The decrease in gray matter volume was possibly because the CSF, because of an increase in CSF, it manifests because there's more sort of dark spaces in a T1 weighted images because the CSF is now eating into those gray matter voxels. It's creating a partial voluming effect, and it's reducing your cortical thickness. So using this multimodal MRI helps you sort of understand what's going on at a much more granular level. Also, if you're starting a project, what we did was we did use an acceleration factor, but we used so therefore we acquired 2 sets of images. And so this allows us to look at reproducibility. We have 2 sets of images. We can take one set, run the analysis, and see if you can reproduce those same findings in a subset of the data. And so you can really help test the reproducibility of your findings. In terms of experimental design, I think what's worthwhile is to make sure that your control group is well matched, give the control group an effort matched task, of course use multimodal MRI data whenever possible, collect 2 sets of data, and there are a host of potential confounds that I've just started thinking about. So in terms of biological rhythms, you want to know about circadian rhythm. There are things like the post-lunch dip. So if you scan someone immediately after lunch, you might get a completely different neural response because of a physiological effect. There are other sort of cycles that people want to be thinking about, like infradian cycles. There are other seasonal cycles. So one study has shown that based on the daylight savings time, there are changes in the hippocampal volume across a group of subjects. And this correlates with how well they do certain tasks. Also, it's worthwhile. I know that it's really hard to control for all of these factors, but if you scan someone at 8 in the morning, chances are they are coffeeed up, and that is going to change your BOLD signal, that's going to change your T1 signal, and it's going to cause issues relative to a subject who is scanned at 4 in the evening or so on. So what I'd like to do is to just get all this data. Just ask people, use a questionnaire, a quick questionnaire. How many cups of water did you drink? How many hours of sleep do you usually get? How many cups of coffee did you have? What was the last time you had a coffee? And so on, right? Get all this data. It might come in handy at some point. So basically, if you can't screen or control for it, measure it or be mindful of it. Also, it's worthwhile to report the methods you use in as much detail as possible. So what I've done so far is, we reviewed the popular MRI methods, went over the limits and the details of scanner-related factors, sequence-related factors, and so on. And I've also sort of walked you through how to overcome some of these limits. And now I'm going to briefly talk about like the future of imaging changes in brain anatomy. I think it's sort of important to so one limitation of MRI is that it has very limited biological specificity. What you're imaging at the core is hydrogen spins. We're not imaging neurons. We're not imaging spines. We are making inferences about certain structures purely based on the spins of the hydrogen nuclei. So the only way we can make certain biological claims is to either use converging MRI modalities. So when you bring in different MRI modalities that are sensitized to different tissue properties, we can make a better assessment of what is changing in terms of the biological correlates. So when you look at something as cortical thickness, because as I've shown you, cortical thickness or gray matter volume can change purely because of other reasons that have nothing to do with gray matter volume. It is just some other tissue property that just like CSF or something that can pervade the tissue, and it can just change your inferences about it. So it's important to sort of be mindful. Even like diffusion, you're looking at the diffusion of water molecules, not the axons or the white matter. It is a proxy for imaging brain structure. So what we need is more robust and quantitative maps of biophysical properties of, so what if, like some of these methods, like T1 weighted images are qualitative images. And so qualitative images are basically pixel intensities rather than sort of measures which have physical units. Diffusion MRI, for instance, is a more quantitative measure because the units is millimeters square per second. It is bound by certain physical constraints, right? So then if you look at fractional anisotropy, you're looking at, or the mean diffusivity, you're looking at millimeters square per second differences between two groups of subjects rather than pixel intensities. And these pixel intensities can be brighter and darker because of other reasons. The other important thing where the future imaging efforts can change our understanding of brain anatomy is validation studies. Now, there is a real lack of relating these MRI correlates to actual biological properties. And so doing this in animal models, relating any of these MRI-related changes to the immunohistochemistry or histological measures is going to be crucial for our ability to sort of relate any MRI change to some tissue property. And finally, one thing that I have would really feel the need for is a way of using powerful statistical methods to sort of bring in these multimodal methods. So if you have the same data in different MRI modalities, so you have T1 weighted images, quantitative T1, quantitative T2, in one sense, they're all related in that they are, so because an MP RAGE is looking at, is based on the T1 weighted contrast, so a quantitative T1 will also be highly related to it. So what are the statistical methods or what are the statistical issues that we need to be mindful when you're like bringing all this data together? How do we control for these things? How do we make proper inferences? So this is something that I think would really help us bring in all these methods and help us understand what's going on, what's changing in the underlying brain structure. Now, as an example, here is one of the more recent advancements that I'm familiar with. So this is, in the diffusion world, this is the standard sort of maps that we produce based on the standard diffusion tensor. You have mean diffusivity, you have fractional anisotropy, and you have the directionally encoded color maps. Now, with much more sophisticated acquisition methods, you can now produce a whole host of different kinds of maps. So what I'm showing you here is based on a fuller characterization of the diffusion displacement profile. And so RTOP is the return to origin probability of a diffusion of a water molecule. So for instance, if in a highly packed system, like let's say in the corpus callosum, right, where there's high degree of myelination, the return to origin probability of a water molecule is going to be very high. And so you can then map them into tissue contrast, and you get these maps which can help you understand like the difference between regions that are highly myelinated versus regions that are which have a sort of different geometry and so on, a different architecture and so on. So you have return to origin probability, you have return to axis probability, and return to plane probabilities by which you can look at the local architecture of white matter. You can also look at other measures like non-Gaussianity in the white matter, in the perpendicular direction, and so on. You get a whole host. And so these are the equivalent of, if you're familiar with the immunohistochemistry world, you have these different stains. And so these are sort of MRI stains of the living human brain, which can, with the proper sort of behavioral correlates, you can look at how behavior changes can correlate with these sort of diffusion MRI properties. So if you're interested in looking this up, there is a review by Hutchinson et al., which sort of details out, and they've also looked at how to validate this in using the ferret brain and so on. So there's a lot of interesting work that's being done in this using this particular technique. So that's pretty much all I have. So thank you all for your attention, and this is work that I did in the Chris Baker's lab as well as Carla Piapoli's lab, and there are a lot of people who have contributed for my understanding of this particular topic and these slides as well. Before I wrap up, these are some reading recommendations. If you're interested in imaging changes in brain anatomy, there are 4 papers that I would highly recommend, and you can download these slides for your reference. And if you have any questions, I'd be happy to address them.
I know you showed that there are differences between AM and PM measurements, but if you didn't, I know that longitudinal aspect of your study involved training, but if you didn't, did you or any other group who didn't train them, you guys look to see if the changes were reversed between PM and the next day's AM, or like were the two current or two?
Yeah, I see what you mean. So I didn't have those slides, but AM to PM, which is just roughly 2 hours apart, you get like statistically significant changes. But 1 week apart, AM to AM or PM to PM, you don't see anything. Without any training. So it seems, just purely just the time of scan seems to have really striking. And this is not the first report of it. It's probably the first report in the context of a training experiment. But people have reported this in terms of diffusion, resting state, and even task-based fMRI. People have reported it, but it's not out there. And I think what we have sort of stumbled upon is probably related to what we think is the sort of lymphatic system itself, because the regions where we see the systematic changes in CSF are highly spatially correlated with where you see lymphatic. Are you familiar with the lymphatic system? This is a system which clears the metabolites in the brain during sleep. But what we are seeing here seems to be the sort of another cycle of this during awake behavior. So there is a slow. So if you imagine the lymphatic system as during sleep, you squeeze like a sponge that's soaked in water. The water extrudes out, and it clears all the metabolites. So now the sponge is squeezed out. It's going to slowly expand, and it's going to reabsorb the water. So what we are looking at, it seems like it's that reabsorption, but it's very, very slow compared to the lymphatic cycle. So it's physiological, and it's quite striking that we can pick it up in MRI. It's just a couple of hours apart. Yeah. Yes.
Thank you. About your methods, a question about the pre-surfer results you had, but it's not yet tried the longitudinal pipeline of pre-surfer?
Yeah, that's the pipeline that we used. Yeah, so this is the making a template using subject-specific templates and so on. But despite that, we found these. You would imagine that if you use a subject-specific template, these changes are going to be like warped and it might be minimized to some extent. But we pick up these structural, so-called structural changes quite dramatically. Yeah.
You mentioned caffeine and caffeine, but do you have any
No, we
data?
didn't have any data, but there are studies which have, well, there's a lot of bold studies. I'm not aware of a diffusion study, but there are, like last HPM, I saw a study that used T1 weighted images and how caffeine changes gray matter volume in subjects. And I mean, this again, I mean, it's very hard to control for this, especially when we are scanning subjects in NIH. You can't really like control and ask them to like don't drink coffee, because if you don't, like, they probably are not going to be great subjects in doing the task. So it is a,
Either way.
yeah, you're damned if you do, you're damned if you don't kind of scenario.
Just one last question.
Yeah.
So just to confirm, so you found that the long-term training, that there was that improved performance associated with less working thickness, but more gray matter volume. Is that right?
Yeah. Exactly. So it's sort of it doesn't add up to, they don't like, and this has been also been reported in the literature. So same data, they run cortical thickness analysis, they get changes like with, musicians, for instance. It correlates with increases in cortical thickness in some areas, but you don't find it in terms of gray matter volume if you run it through the T1 weighted like VBM pipeline. And that's what we're also finding. It does, yeah. So I mean, so what we'd like to do is to then use these areas and define them, and then do an ROI specific analysis in the entire sort of different visits, and then map those changes. But now you're fishing, you're fishing for your effect now. You might find something, but it's that it doesn't show up in the standard analysis just tells you that that there are certain sort of assumptions made in the processing pipeline that pushes certain effects in certain analysis methods and not so in the other methods. But I think, I mean, these things are fairly easy to do now. So if you want to build confidence in your result, I find it useful to actually run it through both of these things and then see, is this all adding up? And it's worthwhile to report it, as you see it. Because part of the replication crisis is because of this, I think, yeah.